So this whole process kind of starts to get repetitive. The ribosome slides down again, the next tRNA molecule comes, it binds to the amino acid that came before it, and then you get this chain of amino acids that starts to form. Okay. So when a stop codon is reached, the whole process of translation ends, and the whole chain of amino acids is released from the ribosome into the cytoplasm of the cell. Okay. So here's this awesome picture, which you guys have. I think it's on your next page, but I'm going to try and explain it with the picture. I'm going to kind of re-explain with the picture. On Monday, I'll have a video clip for you guys that'll help also. Okay, so we'll start with that to refresh. But here's our mRNA. It was made in the nucleus. Through what process? Transcription. Good, makes the mRNA. It left the nucleus and it came out here to the cytoplasm to this lovely blue thing here is the ribosome. Okay, so the ribosome kind of like clamps around the mRNA and it'll scoot down. Okay? And it'll read one codon at a time, so 3 bases at a time. So it read these 3 bases, this codon, and then this anti-codon on the tRNA matched up. This tRNA had this amino acid attached to it. So the rest of the amino acids that came before it are all in a chain up here. So what happens now, the ribosome moved down, reads this codon, this tRNA, this anti-codon will attach, bring this amino acid, and the whole chain is going to attach to that one. So does that make sense? Like this tRNA is not going to have the chain anymore. The whole chain is going to go onto the newest amino acid. This tRNA is now free to go back out into the cell and pick up another amino acid. So they're like all coming in, and it just keeps scooting down and making this huge chain.
So translation is when it slides down?
Translation is when we take the instructions on the mRNA and make a chain of amino acids from it. Think of it like translating a language. You go from Spanish to English. We're going from base language to amino acid language. Okay? So, those amino acid chains become the proteins when they get freed from the ribosome, and then they twist and curl up. I don't know if you guys remember from your video last week, especially the cystic fibrosis section, how cystic fibrosis is caused because the protein in the lung is misshapen, okay, so that it doesn't let salt out of the lung cells. You guys remember seeing that? So the shape of the protein is extremely important. Right? If the bases are wrong in the DNA, they're going to be wrong in the mRNA. If they're wrong in the mRNA, the codon is wrong. If the codon is wrong, the wrong amino acid is brought, which means that the chain of amino acids isn't correct, so the protein could fold the wrong way. Do you guys see why it's like a chain reaction? Okay, so it all starts with the DNA, but it ends up that the protein ends up being incorrect, which is why like one little base can cause Tay-Sachs disease, where that little boy died before he was 3. He had Tay-Sachs because one letter on his DNA was wrong, so the protein, one of the proteins that helps to keep us alive, didn't come out right. Okay, so it's really serious. That was just some shapes of proteins to show you they're complicated. So each protein chain forms the same shape every time it's produced. So we're talking about hemoglobin, like I said earlier, on your red blood cells. Every time hemoglobin is made, it should be the same shape. The proteins become enzymes and cell structures in your body that help you to be alive. Do you guys want to just do the quick check together so we can be done?
Please.
Okay, we're almost done then. You guys are doing awesome. You're making great time. Okay, so let's quick check translation. What does translation do?
Transfers the direction from the mRNA into amino acids.
Good. So we go from the mRNA to a protein. A protein is the same as an amino acid chain. They're the same thing. It's like saying, you know, this collection of Legos is a Lego car. It's, you know what I mean? You're looking at either the pieces or the whole. Good? Okay, where does translation happen? Where does it take place? At the ribosome, which is in the cytoplasm. Good, Brad. a ribosome in the cytoplasm of the cell. Alright, and what is a ribosome made of? Kind of going back to the very beginning. Huh? Nope, they build proteins.
You said in order to make proteins, it has to have proteins.
Right.
So it's kind of made of protein.
No, because RNA. Okay, it's made up of rRNA, which is ribosomal RNA, which is nucleic acid, not a protein. Okay. I know it's the rRNA is ribosomal RNA. So ribosomal RNA makes up the ribosome. It's all the ribosome is a type of RNA in a certain shape. We call it ribosomal RNA. So what does the tRNA bring?
Amino acid.
It brings the amino acids. You guys are doing really well with this. I know it's complicated, but you guys are actually doing a very good job. So, and the tRNA has something that will match with the codons. What is it? Anticodons. And where do we find the codons? What type of RNA? What has the message? The mRNA, yeah. So the tRNA has the anticodons, the mRNA has the codons. The ribosome reads the codons. Okay? Can you guys see it? I'm trying to stay out of the way. Probably blocking everything on my video. Alright, so what kind of bonds do amino acids form? Peptide bonds. Good, Evan. Peptide bonds. What slides down the mRNA from codon to codon? The ribosome. Good, Jake. Everybody watches this video is going to know what class I was in. I keep saying your names. And then when is the amino acid strand released? When we get to what? Good, Brad. Stop codon. Okay, so we reach the stop codon. That's when the whole thing is done. And on Monday, I will show you this video clip. Okay, and then we'll do, after the video clip, we'll do the page that's on the back on Monday. And then we'll start our project, and you guys will have homework on Monday night. Okay? All right. Sounds good. Good job, guys.